I'm Susan Gottesman. I'm representing Lambda Lunch today, which many of you, but maybe not all of you, know is sort of the prokaryotic interest group at NIH. And we have a habit of, one, nominating our favorite microbiologists to come and give these talks, and two, snagging them for a second talk while they're here. So if you like what you hear today, want to hear about an entirely different topic from Roberto, check with me afterwards, but at 11 tomorrow morning we'll be having another talk from Roberto on bacterial farming of microalgae over in Building 37, and I can tell you the room if you don't already know it. So as I said, Lambda Lunch nominated Roberto to come and give this because he's always doing extremely interesting microbiology. So he's been at Harvard Medical School since 1983. As I was looking through some of his publications, I came upon his list of annual review chapters, and there are 6 of them, and they're all on entirely different topics, and that sort of is characteristic of his science. He does beautiful science, opening up new fields that we haven't thought about, gets bored, I guess, or hands them over to the people that were in his lab and goes on to something new. So he was one of the first people to really do molecular analysis of biofilm formation in bacteria, studies on everything from plasmid replication through what you'll hear about today, which is very different. He cares about microbiology broadly. He's been president of ASM, and his plans for the future, and already he's an active participant in the blog on Small Things Considered. And if you're not familiar with this, I recommend you look it up because it has stories constantly about all the fascinating things that microbes do. So I don't want to take any more of his time. I'll remind you that there's a reception after his talk. There'll be questions, and then there'll be a reception in the library sponsored by FAES, which you're all invited to. And now I'll invite Roberto to come and tell us about Brave New Worlds.
Can we have the first slide? Yes, there it is. Very good. So thank you, Susan, for the wonderful introduction, and also thank you to all the Lambda Lunch for the invitation. It's always a pleasure to come to NIH to be part of Lambda Lunch, and in this case, this great honor to give one of these WALS lectures. And I have to admit that it was April 26 when I received the invitation, but that was April 26, 2016, to come here. And I was very pleased to accept, and then I had a whole page, a document that said instructions on choosing a title. And it was very important. It said that make your lecture special, choose a title that will set it aside. So I said, well, let's take Brave New World. I have to confess that I wasn't expecting that a mere year later this would be re-emerging as a classic hitting the New York Times bestseller list for obvious reasons. That was not the intention. I apologize. The intention here was to give some adumbration of the topic, which is just how this new world, really meaning the Americas, is brave in confronting this particular disease that I'm going to tell you about, which has to do with the meeting of old world and new world, and that was the reason. So I wish I had had the foresight to really use it as a term of the times, but maybe it isn't that good. So I will be telling you about what we hope to convince you that we have some reason to believe that this is an event of recent evolution, which I think is what makes this topic quite exciting. And it is an insect-transmitted pathogen, and you're already getting a sense of who the insect is. I'll tell you a lot about this little creature. But before that, I want to tell you sort of very general background about insect-transmitted infectious diseases. And when we think about that, these days, of course, we think that it's very specialized virus-mosquito interactions that lead to such things as Zika, dengue, chikungunya. That's very much in the news, but of course there's plenty of other insect vector illnesses, and perhaps the one that comes to mind immediately to most people who are thinking about global epidemics is portrayed in this painting from Peter Bruegel, The Elder, The Triumph of Death, I don't know how much detail you can get out there, but it's one of my favorite paintings all time. It's at the Prado. If you ever find yourselves in Madrid, please go see it. It's remarkable. It's depicting, it's not clear. We cannot go back and ask Peter what he was really wanting to depict, but in my view, many of us interpret this as the chaos that must have ensued when the Black Death, the medieval Black Death, would come into a town and basically annihilate the population. And so the concept that Yersinia pestis was being transmitted by fleas was one of the biggest causes of really one of the most dramatic losses of life in human history. And so when we think about Yersinia pestis, we know that there have been 3 epidemics. That's based on the record, and they're mapped here. This is from a paper by Mark Achman, 2 papers really. The first one, the Justinian plague, quite early, the 6th century of the current era, and going first to Greece and then spreading out. Then, of course, the Black Death of the medieval period, 1346, that stayed for several hundred years in Europe, killing, some people estimate maybe 100 million people died of this disease. And then the lesser known one that started in Hong Kong in the late 1800s, the 3rd pandemic. Of course, at that time, it didn't affect so many humans, and that's an important point. It became pandemic. But really to remember that this is a disease that affects really rodents. And humans are basically an accident, that the flea bites by mistake, and then there's human-to-human transmission causing this devastation. But there's several things that immediately come to mind. We have a question. So what are these epidemics due to? Where are these pathogens coming from? And one question that many people in evolutionary biology ask is how old are these pathogens? Were they originated in the initial epidemics? And so how old are these pathogens is an important question. And then really one would like to know, for basic reasons, and also to understand how one can follow these epidemics, is how did they initially evolve? This original evolution. And what is interesting about the plague, and wonderful reconstruction from forensic microbiology, is that people have gone back to Bronze Age corpses and look at the teeth, and they find that the Yersinia pestis that had killed those individuals was really basically the same organism that has stayed around. So evolutionarily, this organism could already go and cause a biofilm in the flea so that it would regurgitate. And so the process of infection seemed to have evolved much before, perhaps 5,000 years, perhaps many, many more years before that. So the emergence of the pathogen is one thing. It has affected humans in many ways, but the actual original evolution remains a mystery. And one would like to know, how did these pathogens initially evolve? And the other thing that is really important to recognize is that once a pathogen has evolved, it needs the right population to invade. And once you have a pest and a pathogen, you of course need the right population. And the population needs to be susceptible, and it needs to be in high density. And so the transmission can be fast and effective before the population is decimated. And so that makes sense why during the 1300s the plague would be such a dramatic effect. And so this fact is key. The pests and pathogens can rapidly invade susceptible host populations. And if the populations are genetically lacking in diversity, that really renders a much greater chance of that invasion to be remarkably successful. Right? So you think, okay, that's the human population of the medieval times, matches that perfectly, but that is not the only place where populations of hosts are like that. And I would say that the topic I want to cover today is equally important as human populations, in the sense that it affects human populations indirectly, and that is if we go to agricultural landscapes. And I know that I'm at the NIH, but this is still related to health and food security. And I think at the end that there should be some interesting lessons that can be carried forward, carried across, regardless of who the host is. And this is a wonderful host if you want to do an experimental system, because there's a great ease in playing with this particular host. So in agricultural landscapes today, densities are very high and diversity tends to be very low. So even if you think, okay, this is something that is interesting conceptually, it's also interesting in terms of what might we as humans be susceptible to our food supply being decimated by the invasion of a pathogen. So, for example, we have the case of Phytophthora infestans, the fungus that attacks potato, and of course the cause of the potato late blight disease that is related greatly to the Irish potato famine, a situation where because of the particular social situation, some million people died in the span of a decade, and another million people left the island of Ireland, and to date the population hasn't quite recovered in terms of the numbers. So this is a case of a pathogen affecting the food supply. And you might say, okay, this is just something that happened back in the 19th century. It's not going to repeat itself, but I don't think we can be so certain of that, because we are quite dependent on crops that are lacking in diversity. So we now know that the potato blight also migrated and invaded in Europe through 2 times. This map, also from a paper recently in eLife, Yoshida et al. As you know, potato originated in the Andes and it was introduced to the European continent. So this is the brave old world. The old world was receiving potato, and many countries became highly dependent on the potato. And there was an initial move of the fungus that originated in Mesoamerica, but it met the potato in Ireland and Belgium and in other places. And in Ireland, the socio-political situation was just made the situation so bad that led to so many deaths. But I do think that this isn't just something that you will see in potato. We see it today. We see it today, and we don't always speak of it, but some 10% of our crops are lost due to the fact that we have very homogeneous populations that are highly dense and are being affected by pathogens. So here's the potato blight. Here we have Fusarium affecting corn. This is a fungus that affects wheat, which I think is a particularly susceptible one, because most of the world has a single, pretty much a single variety of wheat that is being utilized. And there, below here, I have 3 examples of bacterial pathogens. Xylella fastidiosa affecting some 100 plants, different species. Appears to be highly evolved. It's been the same in many settings. And then we have Pantoea stuartii. And then this is an interesting one, is the citrus greening disease. And it's fascinating because while we know the insect vector, the pathogen itself is one of these candidatus. Nobody has cultivated yet. So it's an interesting field of microbiology, knowing that there are some bacteria out there we don't know how to cultivate, and some of them are these important pathogens. So I want to tell you about one particular one, disease which is not this, and maybe some of you have heard of it, maybe some of you have had personal experiences with it, and it is a disease which is the bacterial wilt of cucurbits. And so, as the name implies, if you have a plant of pumpkin or squash or cucumbers, it will wilt. It will, no matter how much you water it, it will appear that it hasn't had any water in a long time. Eventually wilts so that it dies. And before it gets too late, I have to tell you that this work came to my lab a few years ago in the hands of a very talented postdoc, Laurie Shapiro, who loves beetles. And Laurie wanted to know more about the bacterial pathogen, and I think it's been a really wonderful, wonderful interaction. So let me first introduce to you the system, the pathogen, the vector, and the ecology, the molecular biology of the disease, if I may, and then we'll go a lot into the evolution. So the organism involved with this is Yersinia tracheiphila. Its name implies that it loves the xylem, and that's exactly what it does. It basically grows in the xylem, which is not a particularly hospitable environment for microbes to grow, and it grows there very well. So much so that it reaches very high densities there, and it clogs basically the arteries of the plant. It is transmitted by a fascinating beetle, which is a complete specialist. This beetle has co-evolved with the cucurbits, so it only eats cucurbits, and it does it very well. So the beetle is, that you already saw one angle of his picture. Here's another one. We call it, it's called Acalimna vitatum, or the famous cucumber striped beetle. So I don't know how many of you grow cucumbers in the summer in this area, but if you do, you probably have come across the cucumber striped beetle. They are very, very common, and they are quite specialized. And so here you can have a healthy plant, and if it gets infected by the bacterium, through the action of the beetle, it can decimate the plant in a matter of 10 days to 2 weeks. And if you now cut a stem of the infected plant, you can see the ooze, that is the mucoid culture, is basically a pure culture of Yersinia tracheiphila growing at very high densities inside the xylem. So that's the pathogen. And I thought I'd show you the effects in not quite real time, but this is a time lapse of 1 week effect in which we inoculate in the laboratory by injecting a certain amount of the bacterium onto the leaves. So you can see, and this, you will see it in your plants in the garden. If it's happening, there's nothing you can do once this happens. So it's if you now look, I'm going to spend a couple of minutes just telling you about the insides of the plant and the insides of the bacterium as it does this, because we have some knowledge of it already. If you look through scanning EM at the xylem, you can see that there's robust growth of this bacteria inside, but they're moving in the xylem, so this has, of course, been emptied through the preparation. If you come close, you can see that the bacteria are there. They're surrounded by exopolysaccharide. I thought I had to bring in something to do with biofilms and polysaccharides, right? so why not? And so, in fact, we do know that this is essential. And I show you now some graphs of survival of mutants that are avirulent that I'm going to briefly mention. And first, the movie, yes, dramatic of how this xylem flows out, oozes out in the, let's see if we get it. Yeah, so this is just only barely sped up. But you cut it and this ooze comes out. Now this is pure culture of bacteria, very high densities. What otherwise you don't usually see anything growing there. It's not like there's a microbiota of the xylem. This thing is usually pretty much lacking in bacteria because it's not that nutritious to begin with. So as I was saying, we have some mutants. I'm going to mention 3. The first one is that this EPS, the exopolysaccharide mutant, doesn't is completely avirulent, and I also will mention briefly 2 others. One is a type 3 secretion. We'll come back to that. That's, of course, the systems that are so well known in plants and animals that are translocating effectors into cells to cause that are important in disease. This is particularly interesting, the expansin mutant. This is a plant gene. It's a plant gene that is required for restructuring cell wall as the plant grows, and it's been well documented that it was horizontally transferred to bacteria. And nobody's really known what they do in bacteria as of now. Just know that they're quite often in many free-living bacteria. And we have here a function important. And when we look at the mutant that is blocked in expansin, they have a very particular phenotype. So first, what I'm showing you here are the number of bacteria, CFUs, that we have 2 days, 6 days, and 12 days after days post-infection. But in the site where we inject, remember here we're injecting with a needle into the petiole, the place where the leaf starts. And I want you to notice that there's some growth of the wild type, and the exopolysaccharide mutant simply doesn't grow at all. It just is completely compromised in its ability to grow. In contrast, both the EPS and the expansin mutant can grow. But if you look at a distal site, another leaf, which is going to also wilt. Of course, there's no growth already in the EPS mutant, but so you don't see it there. But the key is that at distal sites, the type 3 secretion and the expansin mutant both fail to translocate. So they are able to grow perfectly well. And so, for example, the expansin mutant is just making lots and lots of growth, but it gets stuck in the vasculature. It's interesting because it might mean that there is some restructuring of something that is keeping normally bacteria from traversing across this vasculature of the cells. So that is really all I want to tell you about sort of the disease itself in terms of the typical molecular biology of a disease or the pathogenesis, virulence factors. Really what I want to talk about today is disease ecology. What fascinates me, because where do these pathogens come from? How do they evolve? and in what context are they an effective pathogen, etc. So in order to get into those themes, let me just tell you what happens, how does the beetle interact with the bacterium in terms of making this something that happens in the field. And the first thing to recognize is that if you have ever gone to the wild and sampled a wild squash, not a domesticated squash, but a wild squash, they are remarkably bitter. Many squashes remain quite bitter despite our domesticating them. Others are not so bitter. Zucchini, we all eat raw. But some of them are extremely bitter, and that's because they have these compounds, defense compounds, the cucurbitacins. And these are defensive compounds that are inducible. They are these wonderful compounds, and they are really quite deadly. They are deadly to most animals, us included. But that has led to the coevolution of a set of very, very specific herbivores that have specialized in having lunch on these things. And they basically have no competition. And so they have become very much specialist at eating their own kind of plant. So they become very good at doing it. And we've been studying what are the habits of this creature in terms of what food it likes. Does it prefer infected plants? Does it prefer flowers? Does it prefer leaves? And I think it's a really nice story, so let me tell you about that. It turns out that for Erwinia tracheiphila to get into the plant, one way to do it is we inject it. But really, and one of the arguments I will make for it being a recently evolved pathogen, is really a lousy infective agent. If you paint it on the plant, it does nothing. So it must enter the plant through some sort of wound, either on the leaves or in the wounds that are left in the floral nectaries after the beetles have been there to eat. And so both the leaves and the flowers are potential sources of getting infected if the beetle gets in there and is carrying the bacterium and is able to damage the plant. So one question that we ask is, how does that happen? What food does the bacterium prefer? The beetle prefer? Does it like healthy plants, or does it prefer infected plants? And so this, of course, had to be tested, and the results are quite interesting, because not only are these plants very bitter and they have these cucurbitacins as a defense mechanism, they of course are very spiny, and in terms of the size of the beetle, these spines represent an important physical barrier. And so we ask, what do you prefer, little beetle, if we give you a wilted leaf or a normal leaf? And maybe the answer is obvious, what you would choose if you were a beetle, but it had to be done. And so indeed, if we mock infect a leaf that is still healthy, they eat a lot less area than if the plant is wilted. So already think that the beetle is going to prefer to go and eat a leaf that has some bacteria in it because it's wilted. So then we actually think we know the reason for this. It isn't just that it's easy to wilt, it actually, they get attracted. And so what we did next is we went and captured the number of beetles in traps in the field to see if they were going to wilted or healthy plants, and in terms of the leaves, the foliar attraction, and beetle floral attraction, the flowers. And you have nice correspondence. If the plant is healthy or not wilted in an infected plant, in a non-wilted area, they don't go there as much as if the leaves are wilting. And the opposite happens. If the healthy flower is found, they go to it in much more higher frequency than if it's wilted. So they don't seem to like the flowers that are remaining in the sick plants. And why is that? It turns out that we collected volatiles in the field of leaves and flowers. And what we find is that in the flowers, the healthy flowers are putting out volatiles that are attracting the beetle, that thus helping the transmission, because these are healthy flowers. So the beetles go to the healthy flowers, and the same is for the plants' leaves. In the case of the leaves, only the wilting leaves are producing large amounts of these volatiles that we can collect, and therefore the attraction, not only is it easier to eat there, but actually the volatiles are attracting the beetles to go there. So here's general model of the disease as it happens in the field, and that is we propose that the beetles prefer to go to the wilted leaves. There they eat the bacterium along with the leaf, and then they fly, and if there's a healthy plant nearby, they go preferentially to the flower of that. They infect there. Through there, the infection goes systemic. So then the question is, okay, so this is happening, so it must be a very effective system for the bacterium. But it turns out not only is it poor at getting into the plant, it also is poor at getting into the beetle. So again, a highly evolved pathogen that is going to be carried through an insect, you figure has already evolved really great capacity to colonize that particular vector. That seems not to be the case here. So what we did is we fed the beetle on heavily infected leaves for 3 hours, and then we did DNA extraction to quantitate the number of beetles that were effectively colonized along time, either immediately after the feeding, 5 days, or 28 days. And so what we find is very simple. Initially, you get lots of beetles that have the bacterium, but then most of them lose it. So it's not a great colonizer. So I'm going to show you this sped up movie. This is what beetles do in an afternoon after they've had lunch. Or actually, not true. They extend the whole afternoon for their lunch. So this is in a movie. I just want you to watch it, and then I'll comment. I want you to watch it because I find fascinating these little creatures. They're really quite slow. They just mosey around on the leaf. They bite it. But what I want you to notice, and I'm going to run it again so you pay close attention to it, is that they, unlike us that we tend to eat and then wait a little while and then go to the bathroom, they are actually eating and they're pooping at the same time. So we call this, we have very nice name in disease ecology of plants. We don't call this poop, we call it frass. So now you, if you didn't know the word, now you know a new word. I still call it poop. So, so what is happening is in order for the plant to get infected, it must get a hold of a beetle that is eating on the plant, causing some wounds either on the leaf or on the flower, and pooping at the same time into the womb, wound that is now going to give some chance of colonization, of invasion of the plant. So now I want you to see these little droplets. They don't always fall on the wounds, but occasionally they do. And at that point, you can have a productive infection. So you see they're dropping, they're constantly making the little drops, and most of them are not falling in the wounds, but eventually you get some that are, and so you get the plant. So all of this goes to say that if this was a pathogen out there in wild squash, it probably wouldn't do very well because its transmission is poor and its capacity to get into the plant is poor, so it really isn't that great of a pathogen yet. So it's a problem, pathogen, and the reason it's a problem is because our agricultural landscapes are leading to something to be a successful pathogen, even though it has evolved, as of now, inefficient ways of both transmitting and causing the disease. There's yet another thing about this particular pathogen, and that is that it is extremely geographically restricted. So there are cucumbers, there are squashes, there are zucchinis grown all over the world, and this one is only affecting this particular part of the world, the northeast quadrant of the United States. It's the wilt, bacterial wilt of cucurbits is restricted to this area. So again, indicating that this is something that hasn't quite gathered together yet to penetrate widely, even though these agricultural practices are happening all over the world. So what might it be? And I'm not going to offer answers, it's just clues that this is a remarkably recently emerged, it may be a remarkably recently emerged pathogen. So before I continue, and this is where the brave new world and the old world will eventually come to meet, let me tell you a little bit of the natural history of cucurbits. I've just lumped them all together right now, but it's important that we recognize what their natural history is. And cucurbits can include the genus Cucurbita, and in particular the species Pepo, which are all the squashes, pumpkins, zucchinis, and gourds that we know. And they are New World natives. They came, they evolved in the New World. They are quite important culturally. At the time that wheat was being domesticated out in Mesopotamia, the Mesoamericans were beginning to domesticate Cucurbita pepo precursors, presumably initially to use as gourds for vessels, and eventually became food sources. So the first plant to be domesticated in the New World. And I will be showing the zucchini to signify the New World cucurbits, Cucurbita pepo, the squash, pumpkin, zucchini, and gourd family. Then there is the other family of cucurbits, which are the Old World cucurbits. They evolved out here, and they are Cucumis melo and Cucumis sativus, the melons and the cucumbers. Right? And so they are very different plants. They tend to be softer, and I'll use this as the symbol for them. And so the question is, they are cultivated, and they've been cultivated for a long time. They've been domesticated for a long time. And why is it then that we have this restriction to this geographic area of the planet? So we wanted to begin to address this in the laboratory with some experiments, sort of what we would call mesocosm. We grow the plant and we infect it in the lab and see what happens. So we wanted to see if there was perhaps some temperature dependence of the susceptibility to infection of the host. So here we are infecting the plant, not allowing the beetle to do it, but we're infecting the plant with a fair number of cells, 10 to the 4th cells, and we're going to record what happens to them over time. And we're going to use both cucumber and squash. So cucumber to represent the Old World and the squash to represent the New World. And so the temperatures that we're going to be using are meant to simulate average July temperatures in Texas or Massachusetts, because we want to see why are these things not susceptible. Could it be temperature? And so here's the results of this experiment. And we are measuring the percent remaining alive as a function of days after inoculation. And we are inoculating both the Native American squash, or a cucumber, zucchini squash or pumpkins. In this case, we are using squash. And here, the Old World. And so the first thing that you should notice here is cucumber Massachusetts temperature. It's a little bit more, zucchini Massachusetts temperature, a little bit more susceptible. At Texas temperature, it's completely resistant. So the pathogen affecting the native plant at temperatures more like Mesoamerica are as if the pathogen wasn't there, completely avirulent. The same difference in temperature is seen if you grow in Texas, the cucumber, Texas temperature, the cucumber, or the Massachusetts temperature, the cucumber. That Texas temperature tends to be a situation in which the pathogen is less virulent. The most virulent we see in the cucumber, in the Massachusetts. So the key not only is temperature dependence, but to me, the most fascinating result here is the fact, I did have the fact that type 3 secretion may be the molecular reason for this, because type 3 secretion is expressed at higher temperature, at higher levels at low temperature. So it may be part of the temperature regulation for those who are interested in sort of molecular explanation. But to me, the take-home message here is the fact that tracheiphila is more virulent towards the introduced cucumber than native squash. So you see what's happening here. We're going to have the introduction of a plant that has never seen tracheiphila because it's been sort of co-evolving slowly with squashes, perhaps never being virulent, and all of a sudden it has a host and a particular climate that is just propitious. It's basically like the perfect storm. You're bringing in the host at high density that it has never seen, but it seems to really like to invade. So with that in the back of our minds, we set out to try to gain more insight about the evolution of this by doing some comparative genomics, to try to get at the idea of when did this guy really become a pathogen. So we did some sequencing, and we started very humble these days. Of course, you can sequence many larger numbers that I'm going to present, but we started with 6 strains. And the first thing that struck me when we saw them, and maybe because I'm very keen on molecular mechanism, was that of all the Erwinias that we could find that had been characterized, only 2 lacked CRISPRs. And so, in case you have been in Mars for the last 10 years, you probably know what CRISPRs are, but maybe you only know that they're great ways of editing eukaryotic genomes. But if you know what they actually do in bacteria, they are the defensive mechanism to chop away DNA of incoming things such as viruses or horizontally transferred genes, etc. So of the Erwinias, only Erwinia tracheiphila is lacking CRISPR. There's this other one, but this is a single isolate that is not particularly well characterized. So we do not know too much about this one. But all the other ones, as you might expect, have CRISPRs, and maybe those CRISPRs have helped keep those genomes sort of intact. And the genome sizes tend to be in the range of 3.8 to 4 megabases. And if you follow the literature of the current thinking of pathogens, is that they're on a path towards genome reduction. That is, once they become specialized pathogens, they tend to jettison lots of the genes that they don't need because they are able to gain a living interacting with the host. So we normally think of pathogens towards genome reduction. The first thing that we noticed is that tracheiphila, the genomes, were about 1 to 1.2 megabases larger than most other Erwinias that we had characterized, that had been characterized by others. So an expansion of a genome. It was gaining genes. And so when we first had a look at this, I really was, I guess, a bit blown away by what had happened to this genome compared to other Erwinias. So that's summarized here. So the genome, as I said, it's about 1.2 megabases greater than the others. It contains 2 complete prophages. It has a plasmid, but here's what I would like to go through the concentric circles with you. Most of them, they're bleaching out a bit, but the important thing is the number of pseudogenes. So a couple thousand pseudogenes. So maybe it's beginning to go towards genome reduction, but it hasn't gotten there yet. So that was interesting. Then the other thing that we have gotten completely used to, but in this case of a pathogen, perhaps not so much, and that is the wide and even distribution of horizontally transferred genes. And in particular, in terms of phage genomes, we have 2 complete phages that are well recognized, mu-like and an enterobacterial-like phage. And then these blue ones are other intact phages. So lots of phages have been coming in, perhaps not surprisingly, given that it has a lack of CRISPR. Then there's, in light blue, there's incomplete phage regions, or still doubted. And then this one is fascinating because it's our ISs and transposons, and they are shown here in gray, and that's where it's bleaching a bit, but it has several dozen, some 72 different insertion sequences that have jumped in the genome several hundred times. So now you have each one of these sites of insertion, then it's jumped to where that same sequence is inserted elsewhere. So the genome is just riddled with pseudogenes, riddled with phage fragments, and riddled with insertion sequences jumping around. Even the plasmid has several copies of insertion sequences that have jumped around. So this, in many ways, looks like a genome in the making, so much so that when you try to do a synteny alignment with other Erwinias that have high amount of gene content conservation, you get a surprise. And that is that if you align, for example, Amylovora with bilingue, you get a rather good synthetic alignment. Most genes are still in the place where they should be. And you can see that there's been this big translocation that puts these genes that are in this strain over here. they are in this other strain over there. But it's still, the synteny is largely conserved, maybe. But if you look at tracheiphila, it's been obliterated. Right? So because of all of these insertion sequences that have come in, etc. So that was only 6. Oh, there was one other thing I wanted to show here, but it's bleached out. This was 6 strains, and what I want you to barely see here is we have mapped the changes. Even though this genome is all been jumbled, when we look at the 6 isolates, there's only SNPs that differentiate. It's not like there's big differences, big chunks of genes that have come in and out. It's only a few SNPs that are varying along the 6 isolates. But still, we wanted to know, this is just 6, and the collection was all obtained in 2008. So in 2015, we went out to the field and collected many more. We did a very broad collection, and we were actually asking not only about the genes, about the genomes, but we were also asking, can we find this organism infecting wild squashes, say in New Mexico or places like that, or is it really restricted that we cannot find it elsewhere? And so we went out into the field, as I said, in 2015 to build a large strain collection. This is a farm in New Mexico where we spotted some wild squash growing around, so we wanted to collect, so we got close to the fence, and we found this site that's a private property, no trespassing, no hunting, no biologist or biological investigations by anyone. And this is outside the fence, a wild squash. So we collected, we got in the car, and we went away. So that was, but I think it tells us something that I think we all should take seriously as well, that there is an attitude towards biological research that lies in lack of education. And I think we all have a role, especially the older we get, to make sure that that is reversed, especially in certain times. So that's the message there. So we went out, and in fact, we never found Yersinia tracheiphila in any of the samples that we collected from outside the region that is known to be affected by this disease. So if it's there, and we want to find out if it's there in low amounts, does it look at all like the one that we're seeing? Was it present already in Mesoamerica as this plant was beginning to be domesticated, or is it really something that more recently evolved, as its genome might indicate? So, so we did go out in many places. Of course, it's still limited. It's just one person. Really, when I say we, it's Lori Shapiro who's going out there. She's the field ecologist. She collected some 90 strains from cucumbers, from melons, from squashes, from pumpkins, different plants, both from wild growing and from cultivation sections. And she would, of course, be biased, and occasionally she would find a sick plant, etc. So we came up with a nice collection, sequenced all 90 of those genomes, still a very modest by today's standards. But what is clear is that once you begin to enlarge the collection, you begin to see the appearance of what seems to be 3 pathovars. A pathovar that seems to prefer the old world, that is cucumbers and melons, and then 2 different pathovars that can infect both of the cucurbits, either the old world or the new world. These we do not know what to make of. Are they new pathovars in the making, or are they errors in sequence assembly? So I would not make too much of these quite yet. They're singletons. But this looks interesting of the 3 pathovars. Here's an interesting fact, because we now have a very small data set, but we have some strains that were sequenced in 2008 and some strains that were sequenced in 2015. And when we look at the nucleotide diversity that is coming up in pseudogenes, it's and the nucleotide diversity that is coming up in the normal, what we think of them as normal genes, it seems that the frequency of accruing nucleotide diversity is increasing in pseudogenes, again strengthening our idea that these are in fact pseudogenes. So the distribution is, I think, what perhaps the most exciting, because it seems that the pathovars are showing some geographic distribution. It seems that this is continuing to evolve with some sense of where they are being cultivated and who is the plant that is being grown. And I should say that we think at the molecular level we might understand a little bit of this because when we look not at the type-3 secretion system, the type-3 secretion systems that are present in the organism probably most likely come from a predecessor Yersinia tracheiphila, but it's the effectors that have been gained that might be giving rise to this geographic distribution. And so when we look at the list of effectors, most of them are conserved in all three of the pathovars, but some effectors seem to be very pathovar-specific. For example, these only found in the C1, these only found, this found in the C1 and that, but these are missing, and then they are completely missing in the mellow. And then this group here are only in the mellow group, but not the other. So it may indicate already some specializing, some evolution towards specializing of that. So let me then summarize what I've told you by way of using a timeline and the way we are thinking about this. So it's not going to be a linear scale, but some 470 million years ago, here 400 million years ago, the rise of land plants. So we have some plants are coming into land, and around 175 million years ago, Pangea breaks, and now we have the beginning of an old world and a new world, and at that point, we have the beginning of the separate evolution of the squash in a new world with its own herbivore collection, and the cucumbers and melons in the old world with its own collection of herbivores. Some 10,000 years ago, then humans begin to cultivate and domesticate these plants on both sides, in the old world and the new world. But now, some 500 years ago, the old world meets the new world as European settlers arrive in the eastern United States, bringing with them their cucumbers and their melons. And now you have a pathogen in the making, perhaps, that finds a host that had never been there, that is now being planted at high densities, and continues to do so for the next 500 years. And we think that this probably is a time that dates the tracheiphila evolution. We have now gone to herbaria, which have collected dry leaves of wilted cucurbits, and we're going to see if we can actually do some forensic microbiology with plants, see if we can sequence at least pieces of those genomes to see if the evolution that we're seeing may be as recent as just 1 century. So that's in the making. I think it's also important to recognize that this is not just an issue of microbiology, it's an issue of anthropology as well. And if you look at what was the extent of forest when the first settlers arrived in the continental United States, and this is shown in the dark, in 1620, by the beginning of the 20th century, most of the forest had been decimated. I should say that quite nicely, New England is recovering, and the forest is coming back. But you can see how this and the intensive agriculture that ensued with respect to such things as cucurbits would lead to the possible, both the temperature and the generation of the high-density plants to give rise to this, perhaps this geographic distribution. So, where do we go for the future? Can we actually follow tracheiphila evolution in real time? That is now, you know, I never liked it when in job talks people talk about future directions, and here I am telling you about future direction. But actually, there's a message to this one. I think what we need is we need to sequence a lot more isolates, and we need to sequence them in a time series. We need to keep sequencing these isolates of pathogens to watch them evolve in real time. That actually is quite expensive if you want to do it with a highly evolved human pathogen. And what you're going to learn the movement, but you're not going to learn too much about big evolutionary. So I think we have a nice opportunity here. We have a guy who appears to be just emerging, but in a field where there's very little funding to sequence massive amounts. So what are we doing? We're going for citizen science. So what we have started, and it's mostly Lori, she is going to cucurbits that are planted in school gardens. So the American schools have a nice tradition of planting pumpkins. And so we are having this summer already 500 schools will be collecting and we hope to expand that to 5,000 schools eventually. So that's going to be a great resource, not only for sequencing strains, but also for following the biogeography of the beetle, etc., and quite importantly, to expose little children to real research. So that I'm very excited about. A lot of strain collecting, and there's even a great pumpkin project where people can go into the web and notate their samples, their observations, etc. That's all being led by Rob Dunn at UNC, which is where Laurie is now spending half her time as she goes to there and back to Harvard off and on. And she also conducts these science schools in Mexico, where we are trying to trace pre-Columbian squashes and seeing how they might be interacting with this pathogen. So the students here are collecting there as well. So it's an international effort to educate and to carry on with science. And I think it's an important lesson there that sometimes when budgetary constraints are there, you reach out and maybe into more creative ways. And so with that, I'll just put the last slide, which is the acknowledgements of all the people that have interacted with Laurie and us in this project. Laboratories of Naomi Pierce, ours, and the labs of Moraes, Mesher, and Stevenson at Penn State, and now the Rob Dunn lab. And Laurie has been recipient of these fellowships. So with that, I'd like to thank you for your attention. I'll be happy to answer questions.
Hi, Roberto. I wonder if you could comment on the effectors that you are seeing that are specific, and if you can, by looking at their sequences, say anything of where they're coming from or how they're evolving.
So I think it's a great question, and we have not gone down to the nitty-gritty of who's doing what. So be delighted with people who really think about what effectors do to discuss that. But all I can say is that it seems to be very few and discrete sets, but what they actually do, in some cases, I'm sure it's known.
And where they come from? So duplications or DNA from somewhere else? Or?
Yes, so that's actually we do know, and so I can tell you, I'll elaborate on that because I think it's interesting. One of them is a HARP-like type 3 secretion, which is known to be quite important in plants, many different plants. And that seems to come from Erwinia, Amylovora, and other Erwinias. So the machinery to build the type of execution system and some of the effectors. Other effectors seem to be coming from far away. So some of them they are definitely horizontally transferred, and those are the ones that we are putting there, and those are the variable ones. So they seem to be getting them as whole genes, and they are not. So in that regard, they are found elsewhere in the chromosome. Yeah, so they're coming from elsewhere. The other thing I want to say is that there's two systems. The other one seems to be the type of system that is used, for example, in Salmonella for entering eukaryotic cells. And there is no phase that we know of this organism ever entering into the beetle. It's just in the gut. And it doesn't colonize, but we are trying to see. So we know that the mutants that I showed you were avirulent are in the HARP type restriction system. We have not yet mutated the other one to see if it has an effect on the beetle colonization, which is already very poor.
Hi. I'm wondering how do you think about the genetic diversity of the host in terms of the plant, the beetle, and then the Erwinia? I mean, do you think that we need to understand the genetic diversity of the two hosts in terms of the transmission network?
Absolutely, yes. I think so, and I think that's where the evolution is going to be so important, because and the reason for looking at wild squashes, and we would love to go to look for wild cucumbers in the old world, to look at that genetic diversity and how that affects transmission, because the sense that one has, and this is certainly pandemic theory, is that what really kills you is the lack of genetic diversity in the host, because that leads to this remarkable transmission. And it happens that we cultivate plants, our agricultural landscapes are extremely lacking in genetic diversity. And one of the approaches now in terms of not necessarily conservation ecology, but just plague control, is to encourage people to put different varieties in their gardens, right? Just and encourage the hybridization and go back. Maybe your squashes will be a little smaller, but that's okay, right? So maybe they might have more flavor. So I think it's a key question, an absolutely key question. And by going to wild isolates, we want to look at that. But that may be the reason why in the wild squashes in the Southwest we don't find it.
With respect to the resistance of the yellow striped beetle to the Erwinia, is that could that be due to any endosymbiont in the?
Whoa, I never thought about that, and I'm sure it has an endosymbiont, and I do not know. This particular beetle its genome has not been sequenced, and I do not know if people have looked. We have not looked for endosymbionts. But it could easily be that the detoxification is related to an endosymbiont.
Right, because there are lots of examples of the evolution between the endosymbiont of the host and the infectious.
Yes, absolutely, and that could be the answer. Yeah.
Let me ask a
Yeah.
final question. So you showed that the temperature gradient was just the bacteria alone, but what about the beetle? Does that care about temperature?
The beetle is interesting because it has the beetle you can find in the warm temperatures, but the beetle has certainly not been hurt by the fact that the disease is spreading in the Northeast. So that now, in terms of counts, that you find a beetle a lot more in the Northeast because you have more susceptible plants. So the beetle is more successful.
Because it likes the wilted
That's
plants.
right. So it is more successful. So the pathogen clearly is helping the beetle out. So it's not that you don't find it, but the beetle, all its natural history indicates that it was present and it did migrate as the cultivation went up. Whether temperature affects it, I don't know. But certainly you find it in both regions. Yeah.
If there are no further questions, we can continue at the reception in the library. Thank you.
Okay.